All right, ready to dive deep into something pretty cool? We're talking about the immune system today, but not the usual stuff everyone talks about.
No antibodies this time.
Exactly. We're going even deeper,
Think rapid
like
response.
before the rest of the immune system even knows what's going on.
That's where these cells, innate lymphoid cells, come in. ILCs for short.
ILCs, okay, got it. And saw the research you sent mentioned differentiation. To be honest, sounded kind of boring at first.
Oh yeah, it's a word that doesn't exactly scream excitement.
But then, as I read through it,
You got hooked.
totally, it's like a story, how these cells get their specialized jobs, almost like choosing a career path, except, on a cellular level.
And that specialization is crucial for the whole system to work. We call it immune homeostasis. Basically, it's this careful balancing act between fighting off threats, but not going overboard and attacking everything in sight.
Makes sense. You don't want your defenses to be too trigger-happy.
And that's actually a big part of what this research paper we're looking at is all about, how ILC differentiation plays into this whole balance.
So, ILCs, differentiation, got it. Well, let's break it down a bit. The paper talks about these subsets of ILCs, ILC1s, ILC2s, and ILC3s.
Right, 3 main players.
What's the deal with those? What makes each one unique?
Okay, so picture this. ILC1s are like your immune system's SWAT team.
All right, love a good analogy. SWAT team, go on.
They're all about taking care of threats that make it inside your cells. Think viruses,
Oh, yeah.
even some bacteria, and they don't mess around. They use these potent weapons, cytokines we call them, like interferon gamma.
Cytokines? I vaguely remember those from, high school biology.
Basically, they're signaling molecules. Immune cells use them to communicate.
So, the ILC ones are sending out a signal flare.
More like a message, hey, we've got a situation over here.
Okay, got it. So, SWAT team is ILC ones. What about ILC2s? What's their specialty?
All right, so if ILC1s are the SWAT team, ILC2s are more like the allergy response squad.
Oh, interesting. So, when I'm sneezing my head off thanks to pollen.
You got it. They deal with allergens, parasites too, and the cytokines they release are different. Think IL-5, IL-13. Those are the ones that specifically crank up the inflammation in those kinds of scenarios.
Okay, so we've got the SWAT team, we've got the allergy responders. What's left? What do the ILC3s do?
ILC3s, think gut guardians. Seriously, they're all about keeping your gut healthy, which is more important than people realize.
Oh yeah, gut health is huge these days. Everyone's talking about the microbiome.
And ILC3s are key players there. They release IL-17 and IL-22, which, if you want to get technical, help regulate the balance of bacteria in your gut, control inflammation there, and even help with tissue repair.
So basically, they're the construction crew as well as the security guards, making sure everything in the gut runs smoothly.
Pretty much.
It's wild though, right? They all start as ILCs, same basic blueprint. But then, bam, totally different roles.
It is pretty amazing.
What's the secret behind this whole cellular career counseling program? How do they decide which path to take?
Well, it all starts in the bone marrow. There's this cell there, a common lymphoid progenitor, or CLP.
CLP, all right. Another acronym for the collection.
Think of CLPs like stem cells for your immune system. They can become all sorts of different immune cells depending on, well, a lot of things.
So these CLPs are hang out in the bone marrow. Then what? How do they choose ILC1, ILC2, or ILC3? What makes them decide?
That's where things get really interesting. It's these signals, mainly the cytokines we were talking about. They act like specific training programs, guiding each CLP down a certain path.
Like a boot camp for baby immune cell.
Exactly. And once they go through their training, they get these, think of them like specialization certificates. In reality, they're key transcription factors. But basically, these tell the cell which genes to switch on, switch off, and that shapes what the cell ultimately becomes and how it acts. Like you've got TBET for ILC1s, GATA3 for ILC2s, and RORγt for those gut-guarding ILC3s.
So it's like their badges, showing what they're trained to do.
That's a good way to think about it. But it's not just about the signals, the cytokines. The research also talks a lot about the microenvironment.
Ah, the microenvironment. What's that all about? Is that like the training ground where all this happens?
Yeah, kind of. Imagine this. Those CLPs, they get their basic training in the bone marrow, right? Then they're deployed to different parts of the body.
Makes sense. Different parts, different jobs.
And where they end up, that environment, that's the microenvironment. It influences how their training continues, how they specialize even further.
So it's not just what signals they get, but also where they are in the body. Location, location, location.
It's a team effort, really. You've got stromal cells, epithelial cells, even other immune cells, all chipping in to fine-tune these ILCs depending on their location. Like in the gut, there's a lot of IL-23 and IL-1. Those create an environment that's perfect for ILC3 development.
Makes sense, given they're the gut guardians.
Right. It's all very well orchestrated.
It's like different parts of the body have their own specialized ILC academies. So cool.
And then there's another layer to this whole thing. It's called the Notch signaling pathway.
Notch signaling. Okay, I'm intrigued. What's that about?
Well, remember how we talked about cytokines being like messages between cells? Notch signaling is more direct. It's like the cells actually talking face-to-face, influencing each other's choices.
So not just messages in a bottle, but actual conversations.
Pretty much. Like a secret handshake between cells, determining which path they'll take.
So hold on. We've got the cytokine messages, we've got the location, and now these secret handshakes. That's a lot of factors determining an ILC's fate.
It's a complex system, that's for sure.
In notch signaling, it's involved in all the ILC subsets.
It's especially important for ILC2 development. It works with that γ3 transcription factor, the one that makes them specialize in allergies and parasites.
Okay, so multiple signals all working together. Got it. But, the research also mentioned these challenges in future directions.
Oh yeah, there's still a lot we don't know about ILCs.
That makes me think there's got to be more to this story, and I want to hear it. But let's take a quick break, and then we'll come back and unpack those unanswered questions and what the future holds for ILC research. Stick with us.
So one of the big mysteries, one of the things researchers are really trying to figure out right now is this thing called ILC plasticity.
Okay, ILC plasticity, lay it on me. What is that? Can an ILC1 suddenly decide it wants to be an ILC3? Like a midlife career change?
That's a great question. Can these cells actually change their roles? And there's some evidence that, yeah, under certain conditions, they might be able to switch subsets, change their function depending on what their environment's telling them.
Whoa, hold on. So they're not necessarily stuck in one job forever. They can adapt.
It's possible, and it would make sense from an evolutionary standpoint. Think about it. Your body's facing all these different challenges. Viruses, bacteria, allergens, parasites, you name it. Being able to shift gears, adapt to the threat. Huge advantage.
So ILC plasticity is like having a super versatile immune army. They can retrain, specialize on the fly, depending on what they're up against.
Exactly. Like soldiers that can be infantry, medics, engineers, whatever the mission calls for. Immune system adaptability at its finest.
I love it. But what are those triggers? What would make an ILC switch roles like that?
That's what researchers are trying to nail down. It's probably a combo of things, like their initial differentiation, certain cytokines, interactions with other cells, even changes in that tissue microenvironment, all could play a role.
So it's like they get new orders, go through extra training based on the latest intel.
That's a good way to put it. And the exciting part, if we can figure out those triggers, those signals that flip the switch, we might be able to use that knowledge for new therapies.
Okay, now we're talking.
Yeah.
If we could manipulate those signals, could we, make ILCs target cancer cells?
Mm-hmm.
Or maybe dial down their activity in autoimmune diseases where they're attacking healthy tissue?
That's the big dream. Harness the power of ILC plasticity for good. And with all the research going on, we're getting closer to figuring out how.
It all comes back to that balance, right? Immune homeostasis. Not too much, not too little, just the right amount of immune response.
It's a delicate dance, that's for sure. Too much, you get autoimmune problems. Too little, and infections run wild. But if we crack the code on ILCs, how they differentiate, how they function, how they adapt with this plasticity, well, we might be able to hit that sweet spot, that perfect balance for optimal health.
Makes sense. And we've been talking a lot about the gut. But I know ILCs aren't just hanging out in the intestines, right?
Oh no, they're all over the place. Lungs, skin, even fat tissue, strategically positioned throughout the body.
So how do they know where to go? Is there like a cellular GPS that tells an ILC, hey, you're headed to the lungs, not the gut?
Not quite that simple, although that would be cool. It's more like a combination of things. Adhesion molecules, chemokines, those are like signals that attract cells to specific spots, other signaling pathways, a whole guidance system directing those ILCs.
So it's like they've got addresses coded into them, guiding them to the right tissue.
Pretty much. And once they reach their destination, that local microenvironment, remember that, it further shapes their development, their function. Make sure they're ready for whatever that specific location throws at them.
Pre-programmed destination, on-site training. Wow. These cells were incredible.
It really shows you how complex and efficient the immune system is. Always adapting, evolving to keep us safe. And ILCs, they're a key part of that.
I'm blown away. And it's awesome that this research paper we're looking at really emphasizes the need for more research. feels like we're just scratching the surface.
It's an exciting time to be studying ILCs. The more we learn, the more potential we see for treating all sorts of diseases. Imagine personalized immune therapies based on your unique ILC profile.
Now, that's the future of medicine I want to see.
It's out there, waiting to be discovered. So many unanswered questions, but with every new finding, we get closer to unlocking the full power of these incredible cells.
It's mind-boggling. These tiny cells, most people have never even heard of them, could hold the key to treating some of our biggest health challenges.
It just goes to show you, we've always got more to learn about the human body. The immune system especially. There's so much power there.
This deep dive has definitely opened my eyes, and hopefully our listeners too. Thanks so much for walking us through this world of ILCs, this whole differentiation process.
Happy to do it. ILCs are a fascinating area, and I love sharing that with others.
And to our listeners, if you're as intrigued by these cells as we are, make sure to check out the references in our show notes. There's a whole world of research out there.
And hey, you never know. Maybe one of you listening will be the one to make the next big ILC discovery.
That's what I like to hear.
Yeah.
Okay, I think we've covered a lot of ground. Before we wrap up, is there one key takeaway you'd want our listeners to remember about ILCs?
Well, I think the biggest thing is ILCs are essential. They're like the first line of defense, that rapid response team we talked about. And the fact they can specialize, become ILC1s, ILC2s, ILC3s, that's crucial for keeping everything balanced.
Yeah, that balance is key. Fighting off threats, but not going overboard, causing more problems. At first I was like, ILC1, ILC2, ILC3, okay, got it. But the more we talk about it,
There's a lot going on under the surface.
how much complexity, it's amazing.
And that complexity, that's what makes them so fascinating. And it's what makes them so promising for future treatments.
You mentioned manipulating the microenvironment. That really got me thinking, could we really treat diseases that way? By, changing the environment these cells develop in?
It's definitely a possibility. We know the microenvironment plays a huge role in shaping ILCs. So if we can understand exactly how that works, we might be able to use it to our advantage.
So, if we want more ILC3s in the gut, we could tweak the environment there to make it happen.
Exactly. Or think about it this way. What if we could create an environment that encourages ILCs to become like specialized cancer fighters? That could be huge for cancer treatment.
And the flip side, too, right? What if we could tone down the signals that lead to those ILCs involved in autoimmune diseases?
That's the idea. It all comes down to understanding this complex dance between ILCs and their surroundings.
It's pretty incredible when you think about it. These tiny cells, most people didn't even know they exist, could hold the key to treating some of our biggest health problems.
It really highlights how much we still have to learn about the human body. The immune system especially, it's constantly surprising us with its power.
This deep dive has definitely been an eye-opener. I've learned so much, and hopefully our listeners have too. Thanks for guiding us through it all.
My pleasure. ILCs are fascinating. I'm always happy to talk about them.
And for our listeners, if you're as excited about these cells as we are, check out the references in our show notes. There's a ton of research out there just waiting to be explored.
Who knows, maybe one of you will be the one to make the next big breakthrough in ILC research.
That's what it's all about. Keep asking questions, keep learning. Until next time, keep exploring.